Hi, I'm Dr. Dan Rubin with Ad Hoc with the Doc, and today we have an extremely special episode here talking to one of my mentors, Dr. Neil Riordan. Thank you for coming here. It's really special to be able to talk to you about this.
Sure, man.
Long ago we got to work together. You taught me huge amounts of stuff. Helped me learn how to think about oncology and how to approach the whole approach a patient and just to approach the whole ology. And so thank you for that. Taught me a lot about IV vitamin C, about the immune system. And now we're sitting here talking about stem cells, which I know very little about. I know through you most of what I know. And I just really, on this Ad Hoc with the Doc, would love to just talk about what types of stem cells you study, you develop, how you go about harvesting or propagating. I know there are mesenchymal stem cells, and there's a difference of those, and there's different types of stem cells out there. And so first off, how did you become interested in stem cells in general many years ago?
Well, we used to make dendritic cell vaccines, and to make bigger, better dendritic cell vaccines, you needed to start with another kind of stem cell called the HSC, hematopoietic stem cell, which is what you use if you're going to do a bone marrow transplant. In oncology, is probably the largest use of bone marrow transplants. There are some autoimmune diseases that they do, but
so you can get those from the bone marrow.
CD34s can fully restore the blood cell making system in the body. So that's what a bone marrow transplant is. Wipe out the HSCs of a person and give them donor HSCs, and without getting too much into it, that's not what we do. We don't use chemotherapy, and we don't wipe out anybody's immune system. We use the other major group of stem cell in the body, which is called the MSC, mesenchymal stem cell. We just say MSC.
MSC, okay.
Yeah, and so those are found everywhere in your body. They line the blood vessels. Every capillary, the smallest blood vessels in your body, is surrounded by them. And
on the outside?
On the outside, yeah. So actually, they control blood pressure. They control. They're the master controllers of homeostasis, or trying normalcy in the body. And as we age, or as certain illnesses are associated with messed up MSCs, and as we age, we just run out of them. So it's a combination of if you're quite ill for quite some time. Certainly, most autoimmune diseases have a dysfunctional. The MSCs are not working properly.
Is there a way to quote, like talk to them or send them a message? Is there that part of stem cell therapy?
There are tons of crosstalk between all the cells in our body, and one of the main ways besides physical contact is secretions of the cells, and they secrete these small things. So cells this big, they secrete these little exosomes, little microvesicles, and that's a major way of communicating.
Is an exosome like a little envelope packed full of information that they want to deliver to another cell?
Yeah, I don't know if they consciously want to do it, but they do.
Who does? The MSCs make the exosomes.
Yep.
And then they send that somewhere else in the body.
Right. So one of the most interesting things that's ever come out since I've been doing this is the fact that MSCs can triage and deliver mitochondria to somatic cells, and it was done in a inflammatory, highly inflammatory. They put something down into the lungs of animals, and then there's a lot of inflammation, and then the cells. There were dead cells, there were dying cells, and there were healthy cells. So the MSCs were able to triage those cells that had enough mitochondria to make it, and they would leave them alone. The cells that didn't have enough mitochondria to make it, they would leave them alone. And the ones that were salvageable, they would form little microtubules and dump mitochondria into them.
That's absolutely fascinating.
Yeah, and they also bud off these little things called microvesicles, which have are capable of carrying mitochondria, and then the microvesicle is made out of the same membrane that cell, basically the same membrane like any mammalian cell, and then it fuses with the recipient and releases the mitochondria into the cell.
Does it act like a little storehouse for them when they need it, or do they release it generally all
Well,
at once?
normally MSCs aren't acting. They don't actively do that. But
Okay.
in this particular case where they demonstrated it, it was a very inflammatory situation. MSCs have natural ways of seeking out inflammation.
Would this be a way to maybe treat people with mitochondrial disease?
Well, actually, it is. We have we're working on an IND right now. That's investigational new drug application for the treatment of Lee syndrome, which is a mitochondrial disease, and we have a couple of patients that were treated under compassionate use that respond really well to MSC IV therapy. So we don't, mechanistically, we don't know exactly what's going on, but clinically, there's vast improvement.
Well, that sounds like great and wonderful humanitarian work that you're doing. That's a treacherous disease.
Yeah.
And that's a disease, most people don't survive that disease, if I remember right.
I don't think any, most die in childhood.
What is the source of the MSCs in that study, in the compassionate use? Is that autologous, or is that...
It's, no, we're using umbilical cord. So mom has a baby, and the baby's delivered, and the umbilical cord's, tied off and cut, and the baby's taken away. And then mom delivers the afterbirth, which is comprised of the placenta, the amnion, and the umbilical cord. And we take the basically the structural tissue of the umbilical cord and dissociate it. And that's our starting culture of MSCs, and we grow them up in large numbers, and then they're clinically applicable at that point.
And do they survive quite a while in your cultures? Or do you have to preserve them before treatment?
Well, so they're frozen down in a certain special medium that makes it so that when you thaw them, they're still alive. So, all what we do is frozen down, and then in our clinic, we have a GMP syringe prep laboratory.
Okay.
So we have a freezer there. Our manufacturing facility is over by the canal. It's not in the city. And so we manufacture there, and we have regular shipments there. And then we have a team that will then wash the cells, get rid of the cryoprotectant, and suspend them ready for application. And then it's literally built in the middle of our clinic. So it's very convenient.
Wonderful. And the name of your clinic in Panama is what?
The Stem Cell Institute.
Stem Cell
Simple as that. It's Cell Medicine, CELL Medicine.com.
Interesting. And I understand that you've authored now, is it 7 books?
No, I've only authored two books.
Okay.
And no intent to author 7.
Okay, wonderful. Well, I saw it yesterday. I saw your book, and
I think maybe you're thinking it's translated into 7 different languages.
That's what it was.
Yeah.
All right, I'll pick up a copy in a different language. Dr. Riordan, so we're sitting here in Scottsdale, Arizona, and you are in town because we had the dedication last night at Southwest College of Naturopathic Medicine for the Neil Riordan Regenerative Medicine Institute. Thank you for the work that you're doing.
Oh, thanks.
Thank you very much. That's at the Naturopathic Medical School. It's wonderful. It's wonderful to see you again.
Yeah.
Be able to sit down and talk with you. I'd love to talk about some of the mechanisms of action of MSCs, and specifically, I get a lot of questions that people ask me about the use of them clinically. We're here in the United States, obviously, and your main clinic. You have a clinic in Texas.
Mm-hmm.
What's the name of that clinic?
It's called the Riordan Medical Institute.
Okay. Are you using stem cell therapy there?
Yeah, we're using bone marrow.
Okay.
There's a lot of good studies showing bone marrow can augment standard orthopedic conditions, also augment standard spine surgeries and procedures. So we use autologous bone marrow, so that's from the patient.
Okay.
So we have a device that spins it down very quickly so that the patient can be consciously sedated, and we draw the bone marrow, and this machine takes 2.5 minutes, and it's ready to go. And if we're doing a joint, for example, you just do it in the same procedure.
So if they're having like a joint surgery?
Yeah, both surgery and in-office non-surgical
Okay.
Procedures we do. We do a lot more non-surgical than we do surgical. But we have an orthopedic surgeon that works with us. We have a board-certified pain guy, and we have a neurosurgeon that works with us. And so the neurosurgeon, obviously, he's going to surgery. But you can augment that healing process using bone marrow, placing it into where you have soft tissue damage. It's also used for helping to fuse, if you have to do a fusion. And then we also use amnion, which is, it's from the same birth tissue that we get, the umbilical cord products, but it's exempt in the U.S. Amnion, as long as it meets certain criteria. So we're able to use amnion, and we're able to use umbilical cord tissue, but it's not. You can't take that and then grow it and do all the stuff we do in Panama yet. I think ultimately it's going to be very common practice, but there's an exemption for tissue. It's called the 361 under federal regulations, and so that's what we're able to do here. The reason we even moved to Panama is in 2004 they passed a law that allowed it basically banned embryonic stem cell research and treatment.
Okay.
And at the same time, it enabled adult stem cell research and treatment. So it's from that 2004 law that we were able to build our laboratory and get fully licensed by the government. And now things are getting, it's maturing a lot, and the regulatory environment is quite strict, but we're kind of an institution down there, and we employ a lot of people. We work a lot. We collaborate a lot with the other folks down there, and
I imagine you have a lot of people flying in from either out of the country or out of state to come get treated by you, both in Texas and down in Panama.
Right. Yeah,
Wonderful.
in Panama, we probably bring in about, now we're between 200 and 250 patients a month that come from all over the place, and a lot from the U.S., Canada, Australia, mostly English-speaking countries, and then so we have a nucleus that comes from the Arab countries as well. But so we have an economic impact, and our clinic is on the 63rd floor of a 65-story building, and then that building's attached to a Hilton hotel.
Excellent.
And we represent around 30% of the business of the hotel, and we're now we've exceeded our capacity there, so we're building a new clinic at at a hospital facility.
Excellent. Congratulations.
And, thanks. And there's a hospital tower. It's really cool. It's all kind of self-enclosed. It's on a one city block. There's a hospital tower.
Yeah.
State of the art. There's an office tower, we have the top floor of the office tower. There's a hotel tower. There's a movie theater. There are several restaurants. There's a grocery store. So you don't even have to leave.
Wow, you just stay inside the whole time.
Yeah, it's a whole complex. So,
nice.
But since traffic is sometimes challenging, it's really looking forward to that. We're also looking forward to, we have so many patients going up and down the elevator that we're really stuck. So they gave us, that was one of the conditions of us putting a place there, is we get our own elevator, so we don't have to share it with anybody else.
Is there a typical treatment length of time if people were to come to Panama that they could?
Yeah, there is. For rheumatoid arthritis, for multiple sclerosis, for frailty of aging, for heart failure, it's based on basically cell dosing, and cell dosing is based on weight. But for the average person, they need to be treated for between 3 and 4 days. So a typical thing is somebody will fly down on a Sunday. If they've never been there before for the first treatment, then we have to evaluate them and do blood work and do a physical exam and just induct them into the process. And then typically on Tuesday they'll get 40 million cells IV. Wednesday 40 million, Thursday 40 million, and they can go home on from me, but do another physical at the end, and then go. So most people are there for about 5 days.
Wonderful. What about the anti-tumor characteristics of MSCs? Has that been studied?
It has been studied. What's interesting is, and I want to talk to you about this afterwards, but epidemiologically, if you take our patient population, has a very low incidence of cancer, and we've talked to a couple different epidemiologists about doing the math on how many cases of malignancy you should have, but it's very, very rare. And I personally believe that many solid tumors and even some leukemias are, there's certainly a lack of homeostasis going on. There's active immune suppression. There's immune cells don't behave appropriately when there's a clinical cancer. And since MSCs rule all that, or at least have something to do with it, my hunch is that the vast majority of cancers are secondary to either localized or systemic dysfunction or depletion of MSCs.
Because so can you use MSC therapy then potentially to treat somebody with an active malignancy?
Potentially, yeah. I mean, we have, because I'll give you two parts on this. One part is that MSCs contribute to tumor growth and metastases, particularly tumor growth. So they're stromal cells, right? Mm-hmm. In addition to their perivascular, they can become stromal cells.
You mean that the MSCs can be inside of tissues?
In tumors, yeah.
In tumors.
Right.
Do they normally reside in normal tissues?
In every normal tissue.
In every normal tissue.
Right.
I just want to repeat that. Okay.
Every blood vessel. So you have blood vessels pretty much everywhere in your body, and they reside on the blood vessel.
And you have tumors that require blood vessel supply.
Right.
So they're inside of tumors. So it continues. Sorry for that.
Yeah. So there's a lot of trepidation. Obviously, with something relatively new, and when you see that these cells are involved, then it makes you worried, right? Mm-hmm. Well, there's never been a study that showed that these cells were induced tumor formation, but there are several studies showing that the cells in the body participate in tumor growth and expansion. So there's a great study done where they took umbilical cord cells. So there's also a big difference between really young, healthy cells and cells in a body that's been through a lot.
You mean MSCs?
MSCs, yeah, MSCs. So in this particular study, they took MSCs that were grown from the adipose, from the fat tissue of adults, and then they took umbilical cord, the MSCs from umbilical cord, and grew them like we do. And then they took, I can't remember the model right off the top of my head, but the title tells it all. Adipose MSCs promote, and umbilical cord MSCs inhibit tumor growth in blah, blah, blah.
Indeed.
Animal model. Right.
Fascinating.
And probably the most dramatic study was of rat breast cancer, and they used rat umbilical cord, or rat MSCs, umbilical cord, I think, I'm pretty sure, and they grew them up, and then they implant a bunch of tumor cells in the flank, and then that becomes a primary tumor, and that it's a very aggressive thing, and it typically metastasizes, and then the animals that have to be sacrificed. And so you see the tumor growth, and they gave one dose into the tumor, and they gave and another group they gave 3 doses IV, and then they have controls for both of them. And you see the control, the tumor's going like this, and the treated group, didn't matter which way they gave them, it goes up, and then it goes like this, and then back down. So there was no tumor. So the title of that article was something like the original tumors, primary tumors disappeared, and there was no sign of metastases 100 days later.
Wow.
Which is, never seen anything ever do that before.
Wow. The MSCs communicate with the immune system then?
Yeah. So what they concluded in the conclusion of that particular article, which was done at Kansas State University, was that they believed that the cancer stem cells had to have been destroyed or induced to apoptose or necrosed or whatever because of that result. Because typically you have cancer stem cells that then can cause metastases, and they're in their discussion, that was their hypothesis of what happened in that.
But a cancer stem cell can't revert to become a normal MSC-like stem cell and behave appropriately.
Well, I'm not sure. I don't think anybody knows the answer to that. But one of the mechanisms of action is transference via exosomes, normal copies of P53, which is a gene that induces, that basically tells the cell it's okay to go ahead and die.
And so that P53, which is the message that tells the cell that it's okay to die, cancer cells tend to have a damaged copy of P53 or lack it altogether.
Right.
So you're saying that an MSC can say, hey, here's some info you might need, and give it to them.
That's well put, yeah.
Very, very fascinating. What are some of the other projects that you're working on besides your research with Lee syndrome and the clinic? Do you have any other research projects in the works?
Well, we just finished our MS paper, which was a clinical trial of MS patients using MSCs, came out. And the results are very good. The side effects were, if any, mild in some people. Then our autism study is completed, and we're in the throes of finishing touches. We're going to submit that for publication here pretty soon. We're a little bit down the road on our asthma trial. Our frailty trial is going on. Our rheumatoid arthritis is continuing, and our osteoarthritis trial is continuing. The sort of the number next for me, clinical trial-wise in the U.S.
So these are all U.S.-based studies?
No, those are all Panama.
Okay.
And then the U.S., we're going to soon file the IND for the Lee syndrome, but also we're going to do a knee osteoarthritis study using the Wharton's jelly product that we make.
What's that?
In Dallas. Well, we mechanically dissociate the Wharton's jelly, which is the source of the cells that we grow in Panama, but we can't, since it's 361 exempt, we can't grow them or anything like that, and we're applying for an IND to go 351 or, basically you're going for a biologic license at that point for the non-expanded. I think, strategically, FDA is going to be happier with a non-expanded product because there's a lot more hoops that you have to jump through. So it's a minimally manipulated, mechanically dissociated product that has live cells in it that we will, that are quantified and can be, standardized for.
The autism study. Obviously, we can't talk about results, and I'm not asking that, but I'm curious about the study design. Is that something you're able to talk about?
Yeah, it was for a pretty tight group of severe autistic right-handed males between the age of 6 and 12 from the tri-state area, because the guy that recruited, the doctor that recruited him was at NYU, and so he recruited them, and the trial design was 4 treatments over a 9-month period. So time 0, time 1 is 3 months, time 2 is 6 and 9 months out, and then they were followed for 1 year after that.
Excellent. Well, I look forward to those results, and I think probably globally people do. Yeah. Very exciting. Dr. Riordan, Neil, you taught me long ago when we were at the clinic doing immunotherapy for people, you really introduced the notion of tumor as an unhealed wound. And I hold that true today and think about that. And so many of the lab tests that we do and so many of the treatments that we do really attend to that phenomenon and disrupted angiogenesis, et cetera, and how the healing process is disrupted. In terms of that sort of tumor-associated biology, do MSCs play a role in the healing process insofar as that tumors as an unhealed wound.
Yeah, absolutely. I mean, they're really are the key to healing. So they go in first, and you know, when we're young, I'll just use this analogy. Like when you were young, you probably fell down, and you had a friend that fell down and broke his leg or arm, or her leg or arm, and you pretty much don't need to do anything, you know, when you're 14 years old. And when you're 84, you're lucky if it even knits. You know what I mean? Because that's
that's the frailty of aging that you're talking about too.
Well, frailty of aging, but you know, your MSCs go like this with age, and 50, that slope gets real steep, you know. And Arnie Kaplan, my mentor and the guy who named these cells MSCs up in Cleveland, he estimates that from birth until, we used his age at the time because I asked him a question when I was writing my book, the Rising Tide. I was like, Arnie, I know capillary density goes down with time, and you know, with age, and I know that, you know, MSCs live on capillaries, so ergo our total body number of MSCs is going to decline with age. How much is that? I can't find it anywhere in the literature. I spent days trying to find capillary density decline with age, and it doesn't exist. And he said, well, I can tell you my best guess, because he's a musculoskeletal guy, so he's looking at tissues all the time. And he says, in the skin, I can tell you the exact number in skin. Because in a baby, you have all these, you know, there's all this up and down, like almost like looking like villi. So you got this folding of the capillary bed. And then as we age, that folding disappears, and then ultimately it's just flat, and then, you know, the skin gets thin and all the stuff. And he said, we did mine when I was 72, and I'm sitting at 2% of the capillary density of a newborn. Wow. So that's for the skin. So anyway, it's a pretty massive decline. And if that is happening in all the tissues in your body, then then those MSCs aren't around to heal the wound anymore, you know? Absolutely. And if you look at chronic inflammation, then these cells are involved in trying to heal the wound. And if you have chronic inflammation, you're exhausting this pool of them, and this pool is throughout your body, roughly evenly distributed. Your liver has the most, being, you know, very, very having so many blood vessels. Your spinal cord has the least, given that it's we built this wonderful cage to prevent damage to it, and the likelihood of you surviving an insult to it is so low. Mother Nature, or God, didn't waste a whole lot of energy putting blood vessels in there, so just barely enough to support it. And that's why spinal cord injury, we have spinal cord injury trials going on too. Spinal cord injury, all we do is we take cells, young healthy cells that secrete stuff that stimulate regeneration, and we put them in that space. That's all we do. It's not. And let them it go. Ain't rocket science. Right. Let's put it like that. So there was a study where they in Taiwan where they cut spinal cords like that, and they go boink, and then they put cells right in there. They put human umbilical cord MSCs in a fibrin, like, you know, a biologic gel. Yeah. And then sure enough, they sprouted again, right? And that's all that was there.
And they imitated the structure of the tissue?
Well, they reconnected. The neurons reconnected.
Interesting.
So, well, the whole spinal cord reconnected to a certain degree. So all the cells participated in the regenerative process. And then because they were human, they were able to fish, you know, stain the tissue afterwards, after the repair occurred. And they showed there were human cells. Certainly they were still there, but none of them incorporated into the CNS. They were essentially prisoners between fibers.
Got it.
And so they are what heals the body, you know, and you know that's why your liver, you know, you pluck out 80% of your liver and it will regrow.
Interesting, right?
Whereas your spinal cord, you go like that, and it's not very happy.
Fascinating.
Yeah.
Fascinating. So as a clinician, I'm sitting here, I'm listening to your wonderful stories, and I'm wondering, especially regarding the frailty of aging that you're talking about, you start thinking about whether or not there's nutrients that support your stem cells, or there's certain diet strategies. I don't know if these have been figured out, but people ask me that kind of stuff all the time about, what diet, what should I eat? And I'm guessing that there's the general array of nutrients that MSCs respond to and need. And so that's kind of sort of question number one. Is there anything, we're not talking about prescribing something for somebody, but is a good healthy diet reasonable when people are starting to think about MSCs and frailty of aging? Because I've noticed that generally in my practice, and when I practiced with you, there was a commonality of certain older people, usually over the age of about 72, that despite them having a diagnosis of cancer, generally appeared healthier and more vital. And those were the people that generally took B vitamins and garlic from being young adults, and they were into health or they made juice. And so it always struck me. I never connected it between, with MSCs. I don't know if it has anything to do with stem cells. That's part one, and I actually forgot part two. So
yeah. Well, so I'll use one of my father's axioms, Dr. Hughes' axioms. So
I remember your father.
He would, he did this more than once. He would say, and he's giving a lecture, he'd hold up his hand, he said, okay, what's the most important nutrient? And then, of course, he did all the vitamin C stuff, and then people say vitamin C and zinc and magnesium and whatever, And he said, you're all wrong. The most important nutrient is the one you're out of. And it's pretty true. So I think the best thing is to test people, make sure they're not out of something.
Very nice. And
in our autism study, I can tell you, not in our autism study, but in our autism patient pool, I can tell you the ones that get better, they get the mo better. The ones that literally go from being severely autistic to not autistic, almost to a person, they've been to a practitioner that cleaned them up, made sure they didn't have any heavy metals. By the way, we have a cutoff. If your heavy metals, they measure lead and mercury. If they're above a certain level, we don't take you. You have to fix that.
In the trial or at the institute?
Across the board.
Okay.
So the reason is because they're very toxic. Mercury is the most toxic molecule to MSCs on the planet. It's parts per billion that kills them. So, and we have kids with mercury loads coming down. It's like, hey, there's no reason to do this because the likelihood of success is way lower than if you just get your kid in a normal, you get his or her blood level down low enough where it's not going to harm the cell. So, heavy metals are no bueno for MSCs. And then if you take one vital nutrient out of the culture medium, they just quit growing.
Wow.
So it doesn't matter which one. You can take out any one you want, and it doesn't matter which one, they will halt. Some of them more quickly than others. Glutamine is the number one. If you don't have enough glutamine, everything will just fall off a cliff real quick.
Fascinating.
Yeah.
Sorry, that's fascinating to me. All right, last question, because I know we've been talking a long time. It's been a long day. Are there any genomics associated with higher or lower or more efficient or less efficient MSCs or longevity or anything that you're aware of?
There probably are some, but it needs to be looked at further. There's a very big difference in the clinical efficacy of different lots of MSCs. So if there's one thing I think that we figured out that nobody else has, is what markers to look for, and they're not genetic markers. We look at other markers in the cells. We did a big 3-year study, and we were able to figure out what's different, what molecules are underexpressed and overexpressed in highly clinically effective cells. And so now we're able to screen at an early stage and get rid of all those. But when I look at the autism study, the people the clients that we treat, some of them become non-autistic. Some of them go from being severe to mild moderate.
Amazing. Some
of them don't move at all. So I'd really like to know genomically if there's a difference from those super responders and moderate responders, non-responders. My guess is there is, but we haven't done this.
Is it the genome? Well, the MSCs that they're receiving are donor MSCs.
Mm-hmm.
So there could be a difference in the genomics of the donors in association with the genomics of the individual with which they're implanted.
Right. And then to study that, you have to take infinity and square it.
Right. Oh my God, absolutely. Well, any comments before we sign off?
Well, I'm happy to see you, man. It's been a while, and I'm happy to see you're doing so well and doing so many great things with oncology, and just saw you had some students in here, and you're spreading the word and taking care of people, and I'm just really happy to see you and proud of you.
Thank you. Likewise, and thank you for what you're doing and just how you've advanced science and treatment of people. I know what's deep in your heart and how much you enjoy helping people. And I know how, I just know that stems from your family. I've known you a long time. Thank you for what you're doing for the naturopathic profession. And, for what I'm doing, thank you for helping be a mentor of mine and helping to teach me how to think and to really go out there and help people. Thank you. Thanks for being here.
Thanks, man.